This is the
All right. Okay, welcome everybody. Thank you for coming to the interdisciplinary seminar. It's a pleasure to have here today with us Dr. Angelucci from the University of Utah. Dr. Angelucci is an MD, PhD. She got her MD from the University of La Sapienza,
Rome, University of Rome.
where I don't know whether some of you know this or not, where Dr. Lassi is from also, but we won't hold it against her. And she went to MIT to get her PhD in biology and neuroscience, and she's been working on cortical circuits and imaging, and she's now in the Department of Ophthalmology and Visual Sciences at University Medical School of Utah, and she's also a professor in bioengineering. It's a pleasure to have you. Thank you for coming. The floor is yours.
Okay, thank you. Thank you for inviting me. It's a pleasure to be here. This is a really nice place. I didn't here. Before I start, I guess I would like to ask how many people in the audience, and you can look at those in the screen, are actually neuroscientists to get an idea of, how specific. Nobody. Okay. So, I guess I've added a few slides here that are introductory slides, and then hopefully I will introduce all the terms I use. But please, at any time, stop me. This is a small audience. We can do this very easily, and ask me if I'm using any terminology that is familiar to you, and I'd be happy to. I don't have to go through all my slides. I can stop at any time. Okay, so I want to start by thanking my collaborators, the people I've been working with. I mean, most of the work I'll show you today, the modeling work, is done by Paul Breslov in collaboration with Paul Breslov and Laj Babe. Paul is a professor in the mathematics department at the University of Utah, happens to be my husband too, which is useful. And then Jennifer Uchida and Shishu, they're a postdoc and a graduate student in the lab who have done most of the physiological studies I'll be talking today. So, okay. So because this is not a neuroscience audience, I decided that I needed some introductory slide that remind you a little bit about the brain and about the visual pathway. I don't like using that thing, so I'll use this one. So this is a slide. It's a schematic view, top view of the visual pathway. So most of you will probably know, but anyway, the retina, just to remind you, projects to a thalamic nucleus in the middle of the brain called the lateral genicular nucleus, or LGN, and I'll be talking a little bit about this. Projections from the lateral genicular nucleus reach the first cortical stage of visual processing, which is the primary visual cortex, or V1. From V1, there's a number of other visual cortical areas. This is a lateral view schematic of the macaque monkey brain, which is mostly the primary model I'm working on, showing you, these are salsi open, some of the main visual areas that I'll be talking about. Area V1 is here at the back of the brain, is the largest area. V1 projects to area V2, V3 NMT, and so on. And there's a huge portion of the visual, the cerebral cortex is devoted to processing visual information, and right now there have been over 30 visual areas that have been identified in the macaque brain that are devoted to processing various aspects of vision. One important feature of all these areas, and actually the visual pathway, is the concept of the receptive field. This is the idea that individual neurons at each level of this pathway, especially in the early parts of the pathway, respond to the presentation of specific stimuli within a very confined region of the visual world, which is called the receptive field of the neuron. So in this specific case, you can say this is a B1 cell that responds, these are spikes of the neuron over time, responds to a stimulus, in this case a horizontal bar in the receptive field. If you put that same stimulus outside the receptive field, the cell doesn't respond. But if you put a different kinds of stimulus, so for example, a vertical bar here, the cell still doesn't respond. So the receptive fields have spatially discrete, and also they are tuned to respond to specific stimulus features. In V1, one such feature is orientation. So neurons respond best to some orientations, not others. Now, the concept of the classical receptive field and generally speaking, feedforward models of the visual systems have somewhat dominated the idea of how visual processing occurs in vision, in the brain. According to these models, and they really have provided this, the basis for many ideas of how the brain represents object or form. So this a basic concept to these models is that neurons can be, in visual cortex, can be seen as filters or feature detectors, and that the visual system can be viewed as a hierarchy of filters. So in this model, for example, as we go from lower to higher stages, we go from more simple to more complex receptive field properties. And at the basic of these models is that neurons in higher stages, the properties of neurons are built by the orderly convergence of neurons in the early stages. So for example, imagine that this is a B4 cell responding to a complex shape, such a triangle that would be built by the orderly arrangement in space of neurons in, say, for example, in V2 or V1 that have this kind of orientation. Now, however, this is, although this has dominated the field, there's increasing evidence now that the global aspects of a visual stimulus can actually influence the response of neurons. Yeah, hold on. This can influence the response of neurons in early visual areas to the local attributes of the stimulus. Yes?
Since these are the lines, what if there is a curve which your image is not that straight? Then can our brain still separate those things into curved segments, or?
Yes, well, maybe we'll get back to this, yes. There are models of how one can perceive curvature instead of corners. Yeah. But the basic idea is that this is built by smaller units that have simpler receptive field properties, which in somewhat geometric fashion send input to a higher neuron in a larger area. And so on. Now, there's a lot of evidence that there is, for feedforward models, of, that some kind of feedforward processing does occur in the visual system. So, for example, we do know that the receptive field of neurons do get more complex as we go from area B1, say, to area MT, and that the receptive field size gets larger. However, there's also evidence that there are the global to local interactions. So the global attribute of an object can affect the response of neurons with small receptive fields to the local attributes of the visual stimulus. And one fundamental example in visual cortex of these global to local interactions are centers around interaction or contextual effects, which is the title of my talk. What are these? So these are represented schematically here. Think of this as a V1 neuron that is presented with an optimal grading stimulus. So this is oriented at the cell's preferred orientation. Inside its receptive field, you can see that the neuron responds optimally to the stimulus. Now, as I showed you before, the same stimulus outside the receptive field evokes no response, but there is modulation of the response when the center and the surround stimuli are presented together. In this specific case, you can see a suppressive effect to the surround stimulus. Now, this is something that can't easily be explained by feedforward mechanisms and by classical receptive field concepts, because it implies that the surround of a V1 neuron receptive field is influencing somehow the response of the neurons to stimuli in its receptive field. Now, these centers around interactions are being characterized by many laboratories, and there's general agreement that they are orientation specific, and what this means is that the specific sign of the modulator effect, that is whether it is suppressive or facilitatory, depends on the relative orientation in V1 of the stimuli inside and outside the receptive field. However, there's been disagreement among labs about the specific sign of the modulation, meaning under which conditions it's facilitatory or suppressive. Now, at least in part, the reason for this discrepancy can be explained by the fact that different laboratories have used different stimulus configurations and have used different definitions of receptive field center size. So we now know that the size of the receptive field of a visual cortical neuron depends on the way it is measured. So a classical way that a receptive field size has been defined is as the minimum response field, which is usually mapped using small visual stimuli, very tiny visual stimuli moved back and forth inside the receptive field. This is the traditional Hubel and Wiesel method of describing a receptive field. However, it's been shown that another way could be you can use high contrast gratings of increasing diameter, and this would be this is a typical response of a V1 neuron, and you can see that the response increases and then it's suppressed as the stimulus size gets larger. And you can take the stimulus size, define the stimulus size as what I call the summation receptive field at high contrast as the stimulus size at the neuron's peak response. So if you do this, you can see that that region of the receptive field is larger than the minimum response field, and it tends to be about twice as big on average across the population. Now, more recently, it's been shown that if you use a low contrast grading and perform the exact same paradigm, the receptive field is even larger. So you can see that low contrast, the size tuning curve, the peak of the size tuning curve shifts to the right, so the receptive field is even larger. Now, in some studies, the receptive field center was defined as the peak of the high contrast size tuning curve, so the summation receptive field at high contrast, and the surround as the region outside it. Now, in these studies, placement of gratings, large grating stimuli such as this in the surround have been shown to have predominantly suppressive effects on ISO-oriented gratings in the receptive field center. So when this is ISO orientation suppression, that is the stimulus in the receptive field center and in the surround are at the same orientation. And this is regardless of whether the center stimulus is presented at high contrast or a low contrast. Now, in these kinds of studies, when the stimulus presented in the surround is at the orthogonal orientation, so people have observed facilitation. So, and we refer to this as cross-orientation facilitation. So typically, these studies have reported iso-orientation suppression and cross-orientation facilitation. However, okay, before I show you other studies. So these studies that typically model the response of a V1 neuron using these simple statistical models. They are difference or ratio of Gaussians, whereby the receptive field center mechanism is described as an excitatory Gaussian, and the surround mechanism is an inhibitory, a spatially broader inhibitory Gaussian. Now, this model predicts that stimuli in the surround region, in the far surround regions here, can only be suppressive, and this is due to the spatially broader inhibition. However, people have reported that there are facilitatory effects from the surrounds, even at iso orientation. And this has usually happened when the receptive field center is being defined as the minimum response fields, so it's sort of a smaller receptive field. And in these studies, typically people have used Gabor patches or bars, oriented bars, presented inside the minimum response field. And in this specific case, the presentation of co-aligned, so collinear and co-aligned iso-oriented Gabor patches in the surround region have been shown to cause facilitation. And one interpretation of the other camp of these effects is that, well, this may be happening, you may be seeing this facilitation because in effect these stimuli are really involving what the other camp would call still the receptive field center mechanism, and so that's why you see this facilitation. Well, I'm going to show you, if I get to it later in my talk, that in fact we find that even if you use the appropriate visual stimuli, you can find that these facilitatory effects can also arise from the surround, the far surround region, so from this region here. Anyway, so these collinear facilitation effects have been thought to play a role in perceptual counter-integration, which is the mechanism by which our visual system can extract a path see, this is curvature perception, right, of sort of partly co-aligned Gabor patches in the middle of, in a noisy environment. Whereas the suppressive interactions of the kind that I showed you before using these large gratings have been thought to play a role more in the perception and in figure ground segregation and in the perception of occluded surfaces. And this is one example. For example, this is some kind of figure ground segregation based on orientation differences. You can perceive this circle. But also, to give you an example of perceptually how the suppressive interactions do play a role in our vision, is the fact that if I remove the background, the surround stimulus here, you can see that this. So you can see that this the grading is perfectly vertical, but the presence of the surround makes you perceive it as tilted away from the surround. So these are suppressive effects of the surround that operate in vision, and possibly these kinds of neural responses represent the neural correlate of this perceptual phenomena. So a major question in the field is, one is, of course, what are these center-surround interactions for in vision? And a second question is, how are they generated and how do they occur in the visual cortex? What are the pathways and the mechanisms by which neurons acquire these responses? And there's a number of circuits that may be involved. And originally it was proposed that horizontal, long-range V1 horizontal connections are the substrate for these, for center-surround interactions in V1. What are these? Okay, let me first explain this slide. This is a transverse section of cat visual cortex in this case. So the visual cortex is sliced like this, and what it shows is a dominant feature of the visual cortex, which is older cortex, which is a layer structure. Layers are specialized compartments that somehow receive specific inputs and send specific outputs, and also can show different response properties. So in the layer 2-3, there are these cells that are called pyramidal neurons, or excitatory neurons, and in this specific case, this one is an individual neuron that was filled with a tracer, and all of its axonal arbor is filled. These travel within the same lamina, and you see they send these axon arbors that travel long distances, for millimeters within the cortex, and they arise from excitatory neurons, and they contact both excitatory and inhibitory neurons. So because they're long distance, so they can convey information from distant regions of space, they were thought to play a role in these center-surround interactions. Another property of these connections, which is relevant to mediating these center-surround interactions, is their orientation specificity. So I told you that these interactions depend on orientation of the stimulus in the center and the surround. So what you want is a substrate, is a circuit that cares about orientation. And these connections do. So is anybody familiar with optical imaging here? This is a technique that allows us, and I'm not going to get into the detail, but to record the response of populations of neurons when we present, for example, stimuli of different orientations in this case. And what you show, this is a top view of the visual cortex. Actually, this experiment was done by somebody here at Duke University, Boston in the Fitzpatrick lab that I mentioned to you before. And what they did, so what you can see here, different orientations, different colors represent groups of neurons that like the same, respond to the same orientations. And these orientations are mapped in a very regular fashion in cortex, forming what we call orientation maps. Now, if you place a neuronal tracers in one of these orientation domains, so just to remind you, these neuronal tracers work. So this is a neuron. We inject them here at the cell body in cortex, and they are picked up by the dendrites, incorporated into the cell body, and then transported all the way down to the axon. And then we can use, we can section the brain after maybe a week or so, and then we can use antibodies specific to that tracer to reveal these pathways. And this is what you see here in red, is all the terminals, so the boutons that were labeled by a neuronal tracer. And you can see if you inject it in a region that likes a blue orientation, whatever that was, I think it was this orientation in space, you can see that the terminals, the black regions, terminate preferentially in blue. That is, these connections link neurons of similar orientation preference. And they do so along an axis in space that is collinear with the orientation preference of the cells of origin. That is, they will link cells that are collinearly aligned, or the same orientation, are collinearly aligned in this dimension or in that dimension, but not, for example, cells like these that are like the same orientation but are not collinear. So these connections seem to be perfect for doing this counter-integration perception of, or curvature, if you want, and so on. However, more recently, there's been evidence that other pathways are involved. And in particular, these are the feedforward pathway from the LGN to area B1. LGN projects to the input layer of B1, which is layer 4C. And it's been shown in cat and primates that the LGN cells also show these center-surround interactions. So one obvious thing is to think that B1 neurons just simply inherited from the LGN. But I'll show you that that's not entirely the case. We will show you that this will contribute to the B1 surrounds, but it's not the whole story. Then there's feedback pathways, and this is, what are these? These are connections that arise in higher areas, such as B5, B3, B2. They're depicted in blue here. They arise from the upper and lower layers of extrastriate. These areas are also called extrastriate cortex, okay? So, because the striate cortex is B1, and extrastriate is everything that is not B1. So these higher areas can then project back to area B1, and they do this in a very specific fashion in terms of layers. So they target layer 1 as well as layer 2, 3, 4B, and 5, 6. So a number of layers. And one important thing about them is that they also arise from excitatory neurons, and they contact almost exclusively excitatory neurons. This is something that I'll remind you later in the talk. Now, our lab has been one involved in characterizing the functional organization of these feedback connections. And in particular, what we have shown is that these connections, or a subgroup of these feedback connections, are also orientation specific. And so they could also be involved in these center-surround interactions. What this shows you is one of these tracers that injected into RB2. This is a top view of a macaque. In this case, it's a different kind of primate, but it's the same in the macaque. And it shows you how these terminals in layer 1A tend to be sort of diffused, but they form very patchy. You can see these dark regions here in layer 3, in layer 1B, and so on. So in the top layer, these are diffused, but in the layers below 1, they tend to be very clustered. And we in our lab and another lab has shown that this clustering has some relationship to orientation. So what I'm showing you again is the imaging, an optical imaging map. This is of an area V2 where we have injected 2 separate tracers that we can then distinguish. So one is called CTB, and the other one is fluororuby. So the black and the white tracers are placed, targeted to domains, regions of cortex that lie different orientations. And here, this is a map of orientation area B1 with, in black and white, the terminals arising from each injection site. So this would be the feedback connections to B1. And what this shows for each case is the distribution of orientation preferences. In black is under the tracer injection site, and in red is under these terminal fields, boutons in B1. And you can see that they have pretty similar distributions. So what that indicates is that, similar to horizontal connections, these feedback connections tend to link neurons in higher areas and in B1 that have similar orientation preferences. So here I'm moving into more, our data now, and our lab has been concerned with trying to identify what is the relative roles of these connection types, feed-forward, feedback, and horizontal. These are also called lateral sometimes, in generating the surrounds of B1 neurons. And what we did was we performed the studies a while ago in which we basically looked at the spatial extent of each connection type and at the spatial extent of the surround fields in V1. So we asked, how big are these physiological surrounds, and how does this extent correlate with the extent of each connection type? So it's a very basic question. The way we did this was by combining. So we went into a region of V1, and we combined electrophysiological characterization of the responses of neurons at that specific V1 region, and then injected these anatomical tracers so that we could label the connections to that physiologically characterized region. And what we found is that physiologically is that V1 surrounds can be very large. And what this shows you is the radius of these surrounds. And it shows that it can reach up to 13 degrees in diameter. So this is essentially 13 degrees in radius. So this is 26, this is about a screen this big, 22-inch screen that is placed at about 57 centimeters from the animal's eye. So they are huge. When they are actually, if you normalize the size of the surround fields to the individual cell receptive field size, and you do this on a cell-by-cell case, you find a median value of 20 times. So these surround fields, on average, are 20 times bigger than the receptive field of the cell. So you can basically influence the response of a V1 neuron from a huge region of visual space, from very far away. What about the connections? So I'm not going to show you the individual anatomical data, but the summary story of this. What we found is that horizontal connections that were initially proposed to underlie, to be the substrate for these surround fields, are in fact much shorter than people thought in terms of visual field coverage. And these are shown in red. So what I'm showing you here is I represent the receptive field center defined in this way as the white region. In the gray is the extent of the receptive field center measured at low contrast. And this is the surround region. So you can see that the extent of horizontal connections, on average, across the population is coextensive with the peak of the, let's call it the summation receptive field at low contrast. I call this region the near surround. And then what we find instead that feedback connections are much more divergent, and they can match in extent to the largest surround fields that we measured in V1. So based on this, we define two regions in this round. A near surround region where both feedback and horizontal connections can interact to modulate the responses of neurons, and a far surround region outside it, which we think is predominantly dominated by feedback connections. So one question that you could ask is, okay, if horizontal connections are too short, could we then mediate far surround effects using chains, polysynaptic chains of these connections? And so that's when one needs to look at timing. And at the time we were performing these studies, when we initially proposed that feedback may be involved, a number of laboratories were looking at the conduction velocities of horizontal axons versus feedback axons. And the common dogma back then was that, of course, it must take a long time to go from V1, say, to V2 or to V3 and come back, whereas it was assumed that it's much faster to travel within V1 itself. It turned out to be the exact opposite. So it looks like horizontal axons are very slow, partly because they're not myelinated, and feedback axons are 10 times faster than horizontal axons, so that it takes a lot less to go from a higher area back to V1 than to travel long distances within V1. So if you do the right calculations, you can figure out that even using the fastest horizontal axons, which conduct at 0.3 meters per second, it would take 3 to 12 times longer than the measured delay onset of surround modulation for horizontal connections to cover a distance of 13 degrees, which is the largest surround field that we measured. So they would take too long to account for the onset delay of surround modulation, which is very fast. So on the base of propagation speed then and of spatial extent, we propose that the near surround region is mediated by both horizontal and feedback connections, and that the far surround region instead is mediated by feedback connections. What about feedforward connections from the LGN? After we published the studies, laboratories showed that the LGN cells also have this, show this kind of center-surround interactions. So obviously, somehow this must contribute to response of V1 neuron, because if you present a big stimulus, an LGN neuron is suppressed, and of course that must affect the response of a V1 neuron that receives projections from the LGN. So what we did was we performed a similar set of studies as before, where we combined electrophysiological characterization of neurons in a V1 column, in a region of V1, with injections of tracers. And this is shown here. It shows you in white is this black region is the region that is picking up the tracer. Then we go to the LGN. This is a coronal transverse section of the macaque monkey LGN, showing label cells, this dark region, a very narrow column, label cells arising from this. In this specific case, this is 750 microns in diameter, this injection site, which labels a column that is in the parvocellular layers, 350 microns, but even narrower in the magnocellular layers of the LGN. And I don't know if you know the distinction between magno and parvo, but it's not essential. So anyway, what we were able to do is to basically correlate the anatomical spread of these connections from the LGN to V1 with the size, the corresponding visotopic size, and with the size of the receptive field center and surrounding V1 neurons to which they project. And this is the visual field map of these two regions. So what this shows you is that, I'm assuming that you know that the right in an LGN cells are receptive fields that have a center antagonistic surround organization, and they come in two on and off. What we did is we summed up what this represents is the cumulative, what I call the aggregate receptive, classical receptive field of all the LGN neurons labeled here. We found a matched in size the aggregate, basically the cumulative receptive field center size of all the neurons at the V1 injection. What this means is essentially is that the LGN projection, so the feedforward connections, can provide the center and the surround, but this is classical receptive field. It's not these extra classical surrounds that I'm talking about. This can provide a substrate for the receptive field center size of the V1 neurons. And this is just the population results where I'm just here for the whole population. I'm just showing you the ratio of this to this. And you can see that this is close to 1, again, indicating that there is a match in size between the receptive field center size of the LGN cells converging to V1 cells. Essentially, this feedforward pathway is suited to build up the receptive field center size of the V1 neuron, but not the surround. However, at the time that we performed these studies, there were no data on the extraclassical surrounds. So these center-surround interactions I've been talking about, they're also referred to as extraclassical receptive field. And it was shown that, again, as I told you before, that now we know that the LGN cells also show these larger extraclassical surrounds. In fact, this is data now from a different lab, Seniak, in the Callaway's lab at UCSD, where they measured, using the same stimuli was used for V1, they measured the response of LGN neurons, and you can see the same kind of profile. Okay, so they show the same kind of response. And this is the size of the LGN surrounds in different layers of V1. So what they did here is they inactivated V1, LGN sent afferent to V1. They inactivate V1 so that they can actually record specifically from the LGN afferents, and so they can then locate them in the different layers of V1, which is where they terminate. And and they basically found that the surrounds have somewhat. These are their median radius size, and there are some surrounds that can reach up to 2 degrees, and very few only layer 4C alpha that can go up to 4 degrees. So what we did is we added these measurements, their median size and the largest size of LGN surrounds, to our measurements of the anatomical spread of the feedforward projection. And this is what we found. When you use a medium LGN surround sizes and you look at this ratio is still close to 1 or just above 1, which suggests that these surrounds, LGN surrounds, can provide surround fields in V1 that are just a little into the near surround of the V1 neuron. If we use the larger surrounds, For the parvo layers, again, we find the same result. But those few cells that I showed you before that had these very large surrounds could contribute, a surround field in V1 that is 7 or 8 times the size of the receptive field. So they can get into the far surround a little bit, but there is still, I showed you before, and remember if you saw that slide when I told you that in V1, the average size of a surround is 20 times the size of the receptive field. These can provide about 7 or 8 times. So there's still a large fraction of this far surround region that cannot be accounted for by these feedforward connections. So in conclusions, we think that feedforward connections must contribute to, but cannot fully account for the V1 surrounds. And this is due to the narrow spatial spread of thalamocortical afferents, as well as to, I didn't tell you this, but the surrounds in V1 in LGN, are not tuned for orientation. So it is not immediate. It is not easy to see how non-oriented, non-orientation specific surrounds in the LGN could build orientation specific surrounds in V1. So to summarize, I still have what? Do I have 5, maybe? Okay, 10 minutes. Okay. So to summarize this part before I get to the modeling, is our proposal is that the feedforward connections from the that the classical receptive field, so the receptive field of LGN cells, can provide a substrate for the receptive field center size of V1 neurons. The feedback connections from higher areas to V1 provide a substrate for the far surround of V1 neurons, whereas horizontal connections, together with obviously feedback here and the extra classical surround of LGN afferents, provide a substrate for the near surround. So what we did is we generated, then in collaboration with the mathematicians, we generated a biologically, anatomically and electrophysiologically based neural network model of center surround interactions in V1. Before I get to the model, I need to remind you that, remember that all these connections that I told you about, feedforward, feedback, and horizontal, all of them arise from excitatory neurons. But while horizontal axons contact both excitatory and inhibitory neurons, feedback axons contact almost exclusively excitatory neurons. So the question is, how can feedback connections that have a predominantly excitatory influence mediate far surround suppression? If you want suppression, you need inhibition, right? So our proposal is that feedback connections excite pyramidal neurons in the near surround in V1 that then send monosynaptic horizontal connections to excitatory and local inhibitory neurons in the center. So I'll show you this in a minute. Essentially, what we've done is we implemented the circuitry which is diagrammed here into a recurrent network model of V1. And in this model, okay, E and I population of excitatory and inhibitory neurons in area V1 in the center, in the near surround, and in the far surround. Feedforward connections contact in this model only excitatory neurons, as do feedback connections, because I told you that feedback axons only contact excitatory neurons. But horizontal axons, here depicted in red, contact both excitatory and inhibitory neurons. So our idea is that a stimulus in the far surround stimulates V1 neurons in the far surround that send feedforward connections to extrastriate cortical areas, say for example area MT, which sends feedback connections back to the near surround neurons, and then via this EI connections suppresses the neuron in the receptive field center. So in order to count, because I told you before that this region, this near surround region, can have both excitatory and excitatory and inhibitory effects. So in order to account for both excitation and suppression, what we did in the model, we used an assumption in the model, which needs to be biologically confirmed, that the inhibitory neurons, here depicted in gray, have a higher threshold and gain than the local excitatory neurons, here depicted in black, that they control. What this means is that if you stimulate the receptive field center with a weak stimulus, so say with a stimulus of low contrast, the inhibitory neurons are still below threshold, so they're virtually silent, but the excitatory neurons are responsive. Now, here I'm going to show you how this model can account for both eyes orientation, near surround facilitation, and suppression, as seen in the data. So when you present, and I want to remind you, this facilitation, well, maybe I didn't emphasize this, but the facilitation is seen in these effects predominantly when the receptive field center is stimulated with a weak stimulus, or low contrast stimulus. But if you, whereas suppression dominates a high contrast. And our model can account for this. So if you present a stimulus, what I'm depicting is these are the different receptive field center and surround components, with orange areas indicating the regions are active when each network submodule is activated. Okay, so now you're stimulating the receptive field center with a low contrast stimulus. Now the inhibitory neurons are inactive because they have higher threshold, whereas the excitatory neuron is responding. A stimulus in the near surround increases the input to the excitatory neurons, but is still insufficient to bring the inhibitory neurons above threshold, so facilitation occurs. A stimulus in the far surround, instead, increases the input via both feedback and horizontal connections to both inhibitory neurons and excitatory neuron. At this point, the inhibitors reaches threshold, the inhibitory neurons becomes active, and suppression occurs. Now, at high contrast, instead, inhibition here kicks in earlier because the inhibitory neuron threshold is reached for smaller stimulus sizes. Now, 2 more minutes, and then I'm going to end. So one prediction, so in this model, because the far surround uses the same basic mechanism as the near surround, but acts via feedback connections, then this model predicts that we should be able to see either orientation, far surround facilitation, when the center stimulus is stimulated weakly, so a low contrast. As I told you before, this is not something that has been experimentally seen before, and I'll just get to it. So this shows you how this happens. It's a similar concept. So a low contrast is a response to the excitatory neuron. Inhibitory neuron is still silent. Increasing the size of a stimulus in the far surround increases the input to the excitatory neurons up to a stimulus size where the inhibitory neurons response threshold is reached, and then suppression occurs. Whereas this model predicts that at high contrast, at a high contrast, there should be only a stimulus in the far surround only generates suppression because the inhibitory neurons are above threshold to begin with. Now, this far surround facilitation is a model prediction not made by current data. Remember, in the introductory part of my talk, I told you that people have only seen these facilitatory effects from the near surround. And the general concept is that these far surrounds are only suppressive. So we think that part of the reason why people haven't seen this far surround facilitation is because most studies have used fixed size, large gratings of high or low contrast, such as this one, which involve both the near and the far surround. This is a stimulus that in our model, like in the experimental data, generates suppression, both a high contrast and a low contrast. So we tested this model prediction of far surround facilitation using a different kind of visual stimulus. And this stimulus is designed to specifically probe the influence of the feedback mediated far surround and to minimize the stimulation, the involvement of these horizontal connections in the near surround. So what it is, we have a grating in the receptive field center and a stimulus, an annular stimulus in the far surround. The radius of this, the outer radius of the stimulus is 14 degrees, and the inner radius is decreased systematically up to a size that is not smaller than the near surround of the cell. So the near surround is not stimulated, we only stimulate the far surround. And we use 3 different contrast combinations. So you can see here, both center and surround are at high contrast. Here's the center is a low contrast, and here both center and surround are at low contrast. And this is the result. What we do is this is a typical size tuning curve of a V1 cell. This is a different cell. So we're increasing the radius of a stimulus, and we do this at high contrast and a low contrast. And we use this high contrast, the peak of both curves to determine what is the receptive field size of the cell here, and this is the size of the near surround. And this is the response of the same cell to this annulus stimulus that I told you. And the three different colors correspond to the three different combinations of contrast. So this is high contrast, and these are two low contrast conditions. So what we have here is the response of the neuron only to the center stimulus. Okay? And then what we're doing here, we're growing the surround towards the receptive field. And you can see that as more the surround is involved, at low contrast, we see the cell is first facilitated and then suppressed. Whereas at high contrast, we see no facilitation. So this is what, this is what our model predicted. And this is just the population average to show you that we just measure the strength of this facilitatory effect in this way, and in blue it shows that at low contrast, over 60% of our population of cells shows a facilitation strength that is 20% or larger, but no facilitation is seen at high contrast. And I'm going to have to stop here because I'm done. Thanks. Actually, let me just show you.
Are there any questions on the one side? Dr. Chung, are there any questions?
Someone say want to ask a question?
She had answered on the next slide.
Is someone asking now?
Go ahead.
I'm going to put a conclusion thing.
What happened?
See?
Okay.
No questions?
No questions.
All right. Okay, any questions here? Yeah.
So what if our some problem happened to our receptive field? Then say our eye is only sensitive, or our brain is only sensitive to fixed orientation. So what happened to our visual system is that we cannot see things or we cannot understand things.
Sorry, ask the question again. What happens if
there's some problem happens to our receptive field or those filters you talk?
What do you mean problems? Meaning that it
dies,
dies, the neuron dies?
Yeah, they're not able to detect the orientation in the other orientations, but only say in the diagonal orientations. Is that you cannot see things or you cannot understand things?
Well, there's a lot.
Yeah, it's complicated.
Okay, it's a complicated thing because in effect, I mean, if you have, say that you have, and it happens in humans, you have a stroke and you have, say, a big region of V1 that gets killed. Yes, you lose vision in that specific part of the visual field, right? And those are called scotomas. It's like completely. If the loss is small, then the visual system, even in the adult, is plastic enough that some of these blind regions can be taken over by the axons of neurons, essentially, that are next by, and so that you can actually fill in these gaps. And this is been shown also that part of these things happen due to this long-range horizontal connections. But is this what you're asking?
Yeah.
Kind of, because I want to get clear about one point, which is
What
why our brain need a filtering in different orientations and in different scale. Because I was confused at one point is when you're looking at an image, just gave you the image, and by the arrangement of different color and patterns, your human intelligence can force you to connect the points, and you can understand it just not physically by the filterings or the collinear connection stuff. So I don't know why we need those functions.
In other words, your prior is influencing
It's
your
like these are
understanding.
the pre-processing which extracts some feature for your intelligence process. But since your intelligence can also have the same function as what you did by your receptive field, why we still need those stuff?
Do you want to answer this question?
Because you wouldn't have prior knowledge of something if you didn't have the ability to.
You only build your
recognize
prior
pattern.
knowledge having if you have experienced it before.
I mean, why don't we could just we have an imaging system?
Well-
We just have the image, and I think that's quite enough, right?
Well, maybe we can take it offline.
I've got a question though about this. This recognition of patterns and fields and lines, is it invariant to if you tilt your head 90 degrees?
Yes.
So your brain already automatically corrects, and is that correction visible?
So
when you're observing
I mean, of course, the higher you know
Right.
that we have invariant. Well, it actually the answer is no. This is not built in these early areas. In fact, in these early areas, everything is very retinotopic. If you move your eyes, the position. The position of that object on your retina changes completely, and so these are not invariant at all. But eventually, you get to higher levels whereby we were talking about this. So you do build up these invariant representations. How that's done is a big
question.
It's a big question.
And is anybody doing some research on what is the next step after your feature extraction and your receive field? You get stimulated in different orientations and you connect them, and but our brain understands more things than that. So
Those are the higher level.
yeah, what's the higher level process based on the features or the stimulated
So
neural signals?
there's a big gap between, how do we go? So we're at a point where we know, for example, okay, V1 cares a lot about orientations, V1 cares about direction of motion, sort of basic, maybe color, but not so much. When you get to V2, and V4, more complex shapes. When you get to inferotemporal cortex, then you start recognizing big categories of objects. So faces, animals, humans, and so cells become responsive to big categories, irrespective of their position in visual field, whether they're close, they're far. So there's an invariant representation. How do we build that is a big question, right? So there are various models. One is you're doing it in a feedforward manner, but there's lots of problems with feedforward models. Most likely the way we do it is a combination of feedforward mechanisms with top-down mechanisms that act via this feedback. And we know still very little about what the feedback is doing. And I believe that this is the first study that we're doing on trying to characterize functionally the feedback connections. And this is the very early stages. I mean, who knows how it is organized, when you're going from inferotemporal cortex to V4, for example. And these connections are massive. So probably they have something to do with, we're saying today, probably what you're doing, during development, you build some kind of internal representations of categories. And then, the feedforward mechanism somehow is, is when an image comes in, you do some kind of comparison between the input that is coming to inferotemporal cortex and compare that with your developed representative previous experience of images, right? And then possibly there's some evidence that the first thing you do is some kind of global recognition, which is then followed by some filling in the details, and that probably involves feedback connections back to these lower areas that care a lot more about the details, right? Orientations of edges, corners, and all these things, and that's how you fill in the details of the object. But this is still, there's bits of evidence here and there, but it's very speculative.
One more question. Yeah.
The stimuli that you used here kind of directly apply to understanding images, right? 2D images.
Yes.
How
You mean images? What kind of images? Natural images?
Whatever. I mean, the dimension of your problem.
Your patterns are
Yeah.
planar in that sense, right?
So my question is about adding more dimensions when you have a third dimension or you have motion involved. Do you have different kind of stimuli?
Yes. I mean, for example, these center-surrounding interactions have been shown to be, they occur, these contextual effects, they occur not just in V1, they occur pretty much in all areas in which they have been examined. And they are feature-specific. So if you go to MT, for example, in MT is an area that is specialized for the processing of motion, then the neurons there care about discontinuities in motion. So they can detect very well something that is moving in, say, the center is at the optimal direction of motion, and the surround moves in the opposite direction. Those are, those cells are, can detect this discontinuous, and they're suppressed when everything is a big field moving in the same. You can do the same thing with color. Differences in color between center and surround for area B4. But even in B1, there's some more recent studies that are looking at different kinds of feature dimensions. And apparently, yes, neurons are tuned to different attributes of this visual stimulus. So you can build stimuli that are, you can add some feature dimensions to it. You can add motion, you can add color, and still see these interactions become more complex.
Let's thank our speaker again. Very quick question. Has anybody looked at the time response of these things and how the syncing is all
Yes.
is all done within the various areas?